In this video, we are going to go over urinary system. We are going to start with the anatomy. Our urinary system consists of kidneys, ureters, urinary bladder, and urethra. The urine is made in the kidneys. It travels through the ureters to be stored in the urinary bladder, and it is removed from the body through urethra. Let's look at this figure. These are the kidneys. They have outer convex surface and an inner concave surface. The concave medial surface has a hilum which opens into the renal sinus. Upper part of the ureter, it expands into the hilum, and it is known as renal pelvis, and it continues down as the ureters. The ureters from both sides, they open into the bladder. In the bladder, the urine is stored and then it is excreted from the body through the urethra. Now let's look at other structures in this figure. This blue vessel is the inferior vena cava. It is present more on the right side, and aorta, this red vessel, is present more on the left side. Renal artery, it is a branch of aorta, and it supplies the kidneys on both sides. On both sides, the aorta, it gives two branches, one on the left and one on the right. These renal arteries, they go to the kidneys to supply the kidneys. The artery on the right side is longer compared to the renal artery on the left side because the aorta is present more to the left. And this inferior vena cava, the renal veins from the left and the right, they come and they drain in the inferior vena cava. The renal vein on the left side, it is longer compared to the renal vein on the right side because the inferior vena cava, it is present more to the right. Nerves to the kidneys, they travel on the arteries. Along with the arteries, they enter the kidneys at the hilum. This is a cadaver, the same structures we can see here. These are the kidneys, and these are the ureters, these thin tubes you see going to the bladder. This is the bladder, they have opened it. So these are the kidneys, the convex surface, the concave surface, which contains the hilum, and these are the ureters coming from the hilum, and they are going to the bladder. This bladder will open into the urethra. This is the aorta, and this is the inferior vena cava. Our kidneys, they are very important structures. They have very important function in the body. They maintain the body's internal environment. They regulate the total water volume and total solute concentration in the water. They also regulate the ion concentrations in the extracellular fluid, and they are important in long-term management of acid-base balance. They excrete metabolites, wastes, toxins, drugs, and we know they also produce erythropoietin. Erythropoietin is very important in RBC production, and then they also regulate blood pressure. They produce renin. We know we are familiar with renin, angiotensin, aldosterone system, and they also play an important role in activating vitamin D, and they also carry out gluconeogenesis if needed. Kidneys, they are part of our urinary system, like we already discussed in the previous slide. In urinary system, it consists of kidneys, ureters, urinary bladder, and the urethra. Now let's look at the gross anatomy of the kidneys in a little more detail. Our kidneys, they are kidney shaped, right? So this is like we call the shape kidney shape because of the kidneys. And we have a lateral convex surface and an inner concave surface. The inner concave surface, it contains the hilum. Hilum, it opens into the renal sinus, which is occupied by the renal pelvis and also by the arteries and the veins, which are entering the kidneys and leaving the kidneys. And kidneys, they extend from the 12th thoracic vertebra to the 3rd lumbar vertebra, and they are present in the lumbar region. Remember, we did region. There are 9 regions of the abdomen. The kidneys, they are present in the lumbar region. Right kidney, it is slightly lower than the left kidney because the liver, it pushes it down. On the right side, we have liver, which pushes on the right kidney. As a result, the right kidney, it is at a slightly lower level compared to the left kidney. Our kidneys, they are retroperitoneal organs. What does this mean? That means that they lie along the posterior abdominal wall. They are present posteriorly. This is the peritoneal cavity, which is lined by the peritoneum, and the kidneys, they are present between this posterior layer of the peritoneum and the posterior abdominal wall. These are our kidneys. This is the right kidney and this is the left kidney, and we can tell this is the posterior because we can see a vertebra here, and this is the anterior abdominal wall. Posterior part of the kidneys is protected by the ribs. Right kidney is less protected and it is more vulnerable because of the liver. It has been pushed downwards, so it is a kind of more exposed compared to the left kidney. And then our kidneys, they have some coverings. Let's have a closer look at the coverings of the kidney. Let us orient ourselves to this figure. This is a transverse plane. This is telling us that this is a transverse plane. So transverse plane, it divides the body into superior and inferior part. And we can see a vertebra. It means this is the posterior and this is the anterior. Anteriorly, we can see pancreas and stomach and intestines. And this is our aorta, and these are renal arteries. These slit like red streaks going there to each kidney. These are renal arteries, and this is inferior vena cava, and they are these blue streaks, they are the renal veins which are coming to the inferior vena cava. Now let's look at the coverings of the kidney. The outermost covering is the renal fascia. The renal fascia, it is the outermost covering of the kidneys, and it is made up of dense connective tissue. It anchors the kidneys and the adrenal glands which are present on top of the kidneys to the surrounding structures. So the outermost covering of the kidney is the renal fascia, and it encloses the kidneys along with the adrenal glands sitting on top of the kidneys. Inside the renal fascia, we have a renal fat capsule. We have a fat capsule which cushions the kidneys, and it cushions and it is also protective for the kidneys. And inside this fatty capsule, we have a fibrous capsule. So these are three layers of the kidneys. You have to know them. Outermost is the renal fascia, then there is adipose cushion, and then there is fibrous renal capsule. Now let's see what can happen. As we discussed before, the upper part of the kidneys, they are protected by the ribs from the posteriorly. So if someone gets into injury in a car crash, for example, or in any blunt trauma, the lower part of the kidney, it is more vulnerable, and the right kidney, it is more vulnerable compared to the left kidney. And also the renal artery, it can get damaged. When renal artery gets injured, one a symptom is that the patient will pee blood in the urine, so his urine will be red and bloody, and this is an emergency and it needs to be taken care of. Let's look inside the kidney. This is an anteroposterior plane that divides the body into anterior and posterior. This figure is telling us that they have cut the kidney in anteroposterior plane. Inside the kidney, we can see three regions. The renal cortex, this is the outermost, this is the renal cortex. Inside the cortex, we have medulla. Medulla has pyramids. You can see more clearly in the diagrammatic form. These are the pyramids, and inside the medulla, we have renal pelvis. This is the renal pelvis. So the cortex, it dips down in the medulla between the pyramids. These are extensions of the cortex between the pyramids. And the medulla, it contains the pyramids. These pyramids, they have a broad base which is facing towards the cortex, and at the apex is toward the pelvis. At the apex, they are known as papilla. Papilla is the point from where the urine, it seeps into the renal pelvis. Now let's come to the medulla. Let's come to the renal pelvis. The renal pelvis is funnel shaped, and it continues with the ureter. Inside the kidney, it divides into major and minor calyces. There are about 3 major calyces, and then each major calyce it divides into minor calyces. And the minor calyces, they are kind of cup shaped, and they are present around here, around the papilla. They will be like this. They cup the papilla of the pyramid, and this is where the urine it seeps into the minor calyx. Kidney has about 8 lobes. So a pyramid and its surrounding cortical dipping, this makes one lobe of kidney. There are approximately 8 lobes in each kidney. And these pyramids, they appear striped. Why do they appear striped? Because they have alternating renal tubules and capillaries. If you can see in this small figure, they have renal tubules, and then they have capillaries. So because of these parallel lines, they appear striped. Infection of the renal pelvis and the calyces is known as pyelitis. Infection or inflammation of the entire kidney is known as pyelonephritis. And these infections, they are more common in females, and they are caused by the fecal bacteria, because females, they have shorter urinary tract, and it can easily get infected from the fecal bacteria. Swear cases can cause swelling of the kidney and abscess formation. Whenever an inflammation or an infection is surrounded by a wall, we call that abscess has been formed. And pus may fill the renal pelvis. If left untreated, kidney damage can result. And how do we treat pyelonephritis and pyelitis? By giving antibiotics. Now let's look at the blood and the nerve supply of the kidneys. Kidneys have a large blood supply. About 1/4 of the total cardiac output goes to the kidneys every minute. Renal artery, it is a branch of the aorta. So renal artery, it comes from the aorta like we discussed before, and this renal artery, then it divides into segmental artery. There are 5 segmental arteries in each kidney. These segmental arteries, they divide into interlobar arteries. Interlobar, you can see from the name that they are present between the lobes. And then these interlobar arteries, they give rise to arcuate arteries. Arcuate arteries are present over here, over the domes. And then these arcuate arteries, they give rise to the cortical radiate arteries. These cortical radiate arteries are the one which give rise to the glomerulus. What is glomerulus? It is tuft of capillaries. As you know that arterioles, they divide into capillaries. But this is what is special about the kidneys is that usually in the systemic circulation, what happens that arterioles, it divides into capillaries, and then those capillaries, they join to form a venule. But over here there is an afferent arteriole, which means this arteriole is bringing in the blood, and then it is dividing into glomerular capillaries, and these glomerular capillaries then join to form an efferent arteriole. So there is arteriole at both ends. And then this efferent arteriole, then goes into the it turns into the veins via peritubular capillaries or vasa recta. These are two different sets of capillary beds. We will go over here. So there is a peritubular capillary bed, and then there are vasa recta. So these efferent arterioles, they can go drain into either one of these, either in the vasa recta or the peritubular capillaries, and then they make the cortical radiate veins. These over here are the cortical radiate veins, which then join to form the interlobar veins. This vein present between the lobes. Like I said, kidney has around 8 lobes. So these are interlobar veins. These interlobar veins, they join to form the renal vein. This renal vein is going to drain in the inferior vena cava. Now let's go over this figure. This is the renal artery. this it divides into segmental arteries. Okay, so these are segmental arteries which are going to each segment of the kidney, and these segmental arteries, this is one segmental artery, this is another segmental arteries, they give rise to interlobar arteries. These are interlobar arteries which are present between the lobes. Interlobar arteries, these interlobar arteries, they convert into arcuate artery. This arcuate artery present over the dome of the pyramid. This is arcuate artery. These arcuate arteries, they give rise to cortical radiate arteries. These cortical radiate arteries then give rise to afferent arteriole. Now this is special capillary circulation in the kidneys, where at one end we have afferent arteriole, and then at the other end we have efferent arteriole. In the middle we have glomerular capillaries, and then this efferent arteriole, it drains into vasa recta or the peritubular capillaries, and then they make the cortical radiate vein. This is cortical radiate vein which join sorry, this thin one is the cortical radiate vein. This cortical radiate vein, many cortical radiate veins, they join to form the arcuate vein, and the arcuate vein, it makes the interlobar vein, and interlobar veins, they come together and they form the renal vein, which drains into the inferior vena cava. And now what about the nerves? The renal plexus, it travels to the kidney alongside the arteries. And what do these nerves known as the renal plexus they do? They regulate the blood flow by adjusting the diameter of the artery. So if I can make it over here, if I can make a little mess, it will be like this. So the renal plexus, it is coming along the arteries inside the kidneys, and they regulate the blood flow by adjusting the diameter of the arterioles. As we learned in the cardiovascular system, that arterioles, they play a very important role in regulating the blood flow to a capillary bed, to a region. So this renal plexus, they act on the arteriole, and as a result they cause it to constrict or dilate, and it is made up of sympathetic fibers which come from the inferior thoracic and the first lumbar splanchnic nerves. Now let's look at the structure of the nephron. Nephrons, they are the structural and functional units that form urine. What is a functional unit? Smallest physical structure capable of carrying out the function of an organ is known as a functional unit. There are more than 1 million nephrons per kidney. Each nephron has two parts, renal corpuscle and a renal tubule. And what are the functions which nephrons perform? So nephrons, first of all, they create a filtrate from the blood, and then they modify that filtrate. They recover certain substances from that filtrate which the body needs, and then they secrete certain substances into the filtrate which need to be removed from the body. Let's look at the parts of the nephron in this diagram. This is renal corpuscle. It is composed of glomerular capillaries and a capsule. Glomerular capillaries, they are specialized capillaries. Their epithelium is fenestrated endothelium, which makes them very porous, and they are ideal for making a filtrate. And that filtrate, it goes in the glomerular capsule space between the visceral and the parietal layer, and from there it goes to the second part of the nephron, which is the tubular part. This is the tubular part. The first portion is known as proximal convoluted tubule. As you can see, it is very bendy, so it is convoluted. And then this is the thick segment or the descending limb. This is the descending limb. And then this is the loop of Henle. This is the descending limb of the loop of Henle, and then this is the ascending limb of the loop of Henle. And then this is the thick ascending limb. And over here, this is the distal convoluted tubule, and this goes into the collecting duct. So this is the tubular portion of the nephron, and this is the renal corpuscle portion of the nephron. Now let's go over the renal corpuscle in detail. Renal corpuscle, it consists of two parts, the glomerulus, which you can see from here, these capillaries present inside the capsule, and this is a tuft of capillaries, and they are highly porous because they are made up of fenestrated endothelium, and because of this the filtration is easy in these capillaries. And I want you to notice that this is afferent arteriole, which is thicker in diameter, and this is efferent arteriole, which is thinner in diameter. So this glomerulus, it lies between afferent arteriole and efferent arteriole. So this is the special thing about this glomerulus. And then the second part of the renal corpuscle is the capsule. This is the capsule which is surrounding the glomerulus, and it is surrounding it almost all over other than this small opening. And this capsule, it has two layers, a visceral layer which clings to the glomerular capillaries, and then it has a parietal layer. The parietal layer is present on the outside, and this layer does not participate in the filtration. And where the visceral layer, it has special cells which are known as podocytes. Those podocytes have foot processes. Those foot processes, they cling to the capillary basement membrane. The capillary endothelial cells, they are resting on a basement membrane. To that basement membrane, those foot processes, they cling, and between two foot processes there are slits. uh, we will see in the next diagram. So those are known as filtration slits, and because of those filtration slits, it is very easy for the fluid to pass from the capillaries to the capsular space. This is the parietal layer of the glomerular capsule. As you can see, it is made up of thin squamous cells, and this portion, it does not take part in the filtration. And this is the visceral layer of the glomerular capsule. This cell is known as podocyte, and these are its foot processes, and the foot processes are attached to the basement membrane of the capillary. These are capillary endothelial cells, and there are slits between the foot processes which are known as filtration slits. Because of these, the filtration is very easy. And this is known as the filtration membrane. The filtration membrane consists of the fenestrated capillary endothelium, the basement membrane, and the podocyte foot processes with filtration slits. Proximal convoluted tubule, it is the first part of the tubular part of the nephron. It is confined to the cortex. So this is renal corpuscle. This is the proximal convoluted tubule. This is present in the cortex. It is lined by the cuboidal cells. Those cuboidal cells, they have a brush border of microvilli and the luminal surface. These microvilli, they increase the surface area for absorption. Like I said before, the nephron, first it makes the filtrate, then it modifies that filtrate with absorption and secretion. So this is the cuboidal cells of the proximal convoluted tubule with apical brush border of microvilli. After proximal convoluted tubule, there is nephron loop, which is known as loop of Henle. Loop of Henle has a descending limb and an ascending limb. The descending limb, initially in the upper part, it is continuous with the proximal tubule, so it will have cuboidal squamous cells, but then it converts into the thin limb of loop of Henle, thin descending limb, and that thin limb, it is lined by the simple squamous epithelium, as you can see here. Then there is an ascending limb. The initial portion of the ascending limb is thin and it has the simple squamous epithelium, but then it turns into thick ascending limb. The thick ascending limb, it is lined by cuboidal or columnar epithelium. The thick ascending limb, it goes up and around here. It turns into distal convoluted tubule. You can see this is also windy. This is also present in the cortex. So PCT, proximal convoluted tubule, and DCT, distal convoluted tubule, both have C in their names and both are confined to the cortex. This loop of Henle, it is present in the medulla, and it is also lined by the cuboidal cell, but it does not have that prominent brush border of microvilli. Why? Because it is more involved in secretion than in reabsorption, so it does not increase. It does not need the increased surface area provided by the microvilli brush border. Now let's see how we will see it under the microscope, how these things appear under the microscope. So these are the structures which are present in the cortex of the of the kidney. So this is a slide of the renal cortical tissue. So let's see the most prominent thing. This is a glomerulus. This tuft of capillaries is known as glomerulus, and then this is glomerular capsule. This will be the parietal layer, and this will be the visceral layer, and this will be the parietal layer. Between the two layers there is a space which is known as glomerular capsular space, and then this is the squamous epithelium of the parietal layer of glomerular capsule. Now this is a proximal convoluted tubule, because we know in the cortex we will find the renal corpuscle, which consists of the glomerulus and the capsule, and we will find proximal convoluted tubule and distal convoluted tubule. So why are we saying this is proximal convoluted tubule? Because the lumen, it is fuzzy due to the microvilli. And then this clear one, this is the distal convoluted tubule, because the microvilli are not there and we can see the lumen. Okay, let's look at this slide. These yellow things, these are glomerulus, which are specialized capillary beds. Why are they specialized? Because they are between two arterioles. In the systemic circulation, the capillary bed, it is between an arteriole and a venule. But in kidneys, this glomerulus, it is a capillary bed which is present between afferent arteriole and efferent arteriole, and the endothelium in the glomerular capillaries, it is fenestrated endothelium. That is why it is very porous and it allows filtration to happen. And now it has an afferent arteriole. Afferent arteriole is wider, and efferent arteriole, it is thin compared to the afferent arteriole. Now let's go over the renal tubule. Now we are here. We already went over all these parts. Now we are in the renal tubule and the collecting duct. Over here we find two types of cells. There are principal cells, which are sparse and they have short microvilli. What is their function? They maintain water and sodium balance. Then we have intercalated cells. Intercalated cells, they are also cuboidal, and they have abundant microvilli. Like you can see on the top, they have abundant microvilli, and there are two types of intercalated cells, A and B, and they both help in maintaining acid-base balance. Now these collecting ducts, they receive the filtrate from many nephrons. So one collecting duct is going to receive filtrate from a number of nephrons, and they run through the medullary pyramids, and they give the pyramids their striped appearance. And then these ducts fuse together to deliver urine through the papilla into the minor calyces. Remember, the apex of the pyramid in the medulla is known as papilla. Under those papilla, the minor calyces are present like cups, and the urine, so it seeps from the papilla into those minor calyces. Let's revise everything we read in this slide. A nephron has two parts, a renal corpuscle and a tubular part. This is the renal corpuscle. It is made up of a tuft of glomerular capillaries, which are specialized capillaries. Their epithelium is fenestrated epithelium, which makes them very porous. The rest on a basement membrane. This tuft is surrounded by a capsule, which is known as glomerular capsule. Glomerular capsule has two layers, a parietal layer, which is this does not participate in the filtration, and then it has a visceral layer. The visceral layer, it is stepped to the capillary basement membrane. The visceral layer has a special cell which is known as podocyte. Podocyte has foot processes. Between the foot processes, there are filtration slits, and these foot processes, they attach to the basement membrane of the capillary epithelium, and on the other side these are capillary endothelial cells, and this is our filtration membrane. And now this is about the renal corpuscle. Now the renal corpuscle, it makes the filtrate, and that filtrate, it goes into the tubular part of the nephron. Tubular part of the nephron is the PCT, or the proximal convoluted tubule. This is present in the cortex. As you can see, this is present in the cortex, and it is lined by cuboidal cells which have apical microvilli, and they also have abundant mitochondria. Why abundant mitochondria? Because many of the substances which are reabsorbed from the filtrate, they need to be transported into the blood using energy. So these mitochondria are there because they provide energy. And you have to notice that this border is highly enfolded basolateral membrane it has. So this is our proximal convoluted tubule, and it continues into the loop of Henle. The upper portion of the loop of the Henle is continuous with the PCT, and it has cuboidal epithelium. But from here it is thin descending limb. The thin descending limb of the of the loop of the Henle, it continues into the thick ascending limb, and then it changes into thick ascending limb. The thin descending and the ascending limbs of the loop of Henle, they are lined by the thin squamous cell. You can see over here in this box. And then this changes into the thick ascending limb. The thick ascending limb, it has cuboidal and columnar cells, and this thick ascending limb, it goes up, and this changes into distal convoluted tubule. This distal convoluted tubule, it is also present in the cortex, and this has cuboidal cells. But notice the difference from the PCT is that its apical surface does not have that many microvilli, because this portion is basically involved in secretion and not in absorption. In the proximal convoluted tubule, these microvilli, they were helping in increasing the surface area for absorption. Now in DCT, we need there is secretion. This is the portion which is mainly responsible for secretion, so that it does not have many microvilli, and then but it does have mitochondria, and the cells are cuboidal. Then this distal convoluted tubule, it ends into the renal tubule and the collecting duct. In this portion we have two types of cells. We have principal cells which are involved in the sodium and water absorption, and then we have intercalated cells. These intercalated cells are of two types, A and B, and they will play an important role in managing the pH of the blood. So this is our nephron. We discussed the basic structure of the nephron. Now let's see how many types of nephrons do we have. We have two types of nephrons. The cortical nephrons, the name is telling you that they stay in the cortex. They make about 85% of the nephrons, and they are almost entirely in the cortex. Maybe a little part of the loop of Henle will be dipping in the medulla, but mostly they are in the cortex. Then we have another type of nephrons which are known as juxtamedullary nephrons. Juxta means next to, so they are next to the medulla. These are long nephrons, and their loops, the loops of Henle, they go deep into the medulla, and the ascending limbs have thick and thin segments, and they are very important in the production of concentrated urine. So they make the urine concentrated. So this is our cortical nephron. It is present in the cortex. It is only going a little bit into the medulla, as you can see, and this is our juxtamedullary nephron. Its loop of Henle, it is going deep into the medulla. Over here you can see the loop of Henle, it is shorter. So now while we are here, I want to say another point, So this cortical nephron, it has peritubular capillary network, and then this juxtamedullary, it has vasa recta. So these capillaries, they are known as vasa recta. Vasa recta, you have heard this term before. Vasa recta are the vessels which provide nutrition to an organ. So this juxtamedullary, it has vasa recta network around it, whereas this cortical nephron, it has this network. These are known as peritubular capillaries. So the same point which I just mentioned. So the renal tubules are associated with two capillary beds. Okay. So one is the glomerulus, which we know is the specialized capillary bed between afferent arteriole and efferent arteriole. And then there is peritubular capillaries. Peritubular capillaries, they are present around the cortical nephron, and they arise from the efferent arteriole, right? The glomerulus, it is arising from the afferent arteriole, but peritubular capillaries, this bed, it is arising from the efferent arteriole. Then the juxtamedullary nephrons are associated with vasa recta. These vasa recta are also coming from the efferent arteriole. Now let's see in detail. So glomerulus, the first capillary bed is the glomerulus. These are specialized capillaries for filtration. They are different from other capillary beds because they are fed and drained by arterioles. There is an afferent arteriole which is wider in thickness, and then there is efferent arteriole which is narrower compared to the afferent. In this diagram, the difference is not prominent. Okay, so the afferent arteriole, it arises from the cortical radiate arteries, and efferent feed into either peritubular capillaries or the vasa recta. Like I said before, that these efferent arterioles, where do they go? Either they make peritubular tubular capillaries if this is associated with the cortical nephron, or they make vasa recta, they drain into vasa recta if it is a juxtaglomerular nephron. And blood pressure in the glomerulus is high. Why? Because it has arterioles on both sides, and arterioles, we know, they are high resistance vessels. And the other point is that the diameter of efferent arteriole, it is narrower compared to the afferent arteriole. So this also increases the resistance in this system. So this glomerulus, it has high pressure compared to the systemic duct of capillaries, and that high pressure, it aids in the filtration, it aids in the movement of filtrate formation, movement of water or the plasma from the capillaries into the capsular space. Now our second bed is peritubular capillaries. Peritubular capillaries, they come from the efferent arterioles. They are low pressure capillaries because their function is to reabsorb the water and the electrolytes from the filtrate. The glomerular capillaries, they are high pressure capillaries because it suits their function. They have to make the filtrate. They have to push the fluid out of the capillaries into the capsule, whereas these peritubular capillaries, they are present around the tubules, and their function is to reabsorb the substance which is needed by the body. So for that reason, it is good for them to be low pressure, and then they cling to the adjacent renal tubules in the cortex, and they empty into venules. So for the cortical nephrons, we have peritubular capillaries, and for the juxtamedullary nephrons, we have vasa recta. They are long, thin-walled vessels parallel to long nephron loops of juxtamedullary nephrons. They also arise from the efferent arterioles serving the juxtamedullary nephrons. And so instead of peritubular capillaries, these nephrons, they have vasa recta, and they function in the formation of concentrated urine. The loop of Henle and the associated vasa recta, they are very important to concentrate the urine. This brings us again to this figure. So now we can just see this is our cortical nephron, right? And this is the peritubular capillary network. and this is our juxtamedullary nephron. These are the vasa recta, and as other point, and this is our third uh, capillary network, which is the glomerulus. So glomerulus is present in all the nephrons, and whereas peritubular is present with the cortical nephrons, and vasa recta it is present with the juxtamedullary. There is a specialized complex present in each nephron. It is known as juxtaglomerular complex. Juxta means close to. And where is it present? It is present in the upper portion, in the distal portion of the ascending limb of nephron loop. So when the ascending limb, just imagine this is the ascending limb, it is coming up, and when at the spot where it is close to the glomerulus, juxtaglomerular. So just imagine this is glomerulus, where it is close to the glomerulus. Over here at this spot, certain changes they happen between the in the epithelium of this ascending limb and the epithelium of the capillary. There are certain specialized changes. We will go over them in a little while. So this is known as juxtaglomerular complex. And what are the parts involved? From the side of the tubule, it is the ascending limb of the nephron loop, and from the capillary side, it is the afferent arteriole. Sometimes it can be efferent, but commonly it is afferent arteriole from the side of from the blood side, and from the tubular side, the nephron side, it is the ascending limb. This complex, it contains some specialized cells. So one type of cells, they are known as macula densa. They are tall, closely packed cells of the ascending limb, okay, and they contain the chemoreceptors that sense sodium chloride content of the filtrate. Then other type of cell population, they are the granular cells. They are also known as juxtaglomerular or GJ cells. They are enlarged smooth muscle cells of the arteriole, and they act as mechanoreceptors to sense blood pressure in afferent arteriole. And these cells, they contain the secretory granules that contain renin. Then there is a third type of cells present in juxtaglomerular complex. These are known as mesangial cells. What are these extraglomerular mesangial cells? They are located between the arteriole and the tubule cells. They are interconnected with gap junctions, and they may pass signals between the macula densa and the granular cells. This is the ascending limb. This is the thick ascending limb. Let me trace it over here. This is the renal corpuscle consisting of the glomerulus and the capsule. This is the proximal convoluted tubule, and this is the descending limb. This is the thin loop of Henle. This is the ascending limb, and this is where the ascending limb, it is coming in contact with the afferent arteriole. So this is the spot where the juxtaglomerular apparatus is going to be formed. And this thick ascending limb will soon convert into distal convoluted tubule. So in some books you might even read where the distal convoluted tubule is. So now this is the thick ascending limb. It is coming up over here, and it is in close proximity to the glomerulus, juxtaglomerular, because this complex is here, and this is the afferent arteriole. Now these cells which are in contact with this portion, they have specialized, and they are not tall specialized cells. They are known as macula densa cells of the ascending limb of nephron loop. These cells, they are sensitive to the concentration of sodium chloride in the filtrate which is coming in the ascending limb. Okay, so they can detect the concentration of the sodium chloride in the ascending limb. So you can say they are chemoreceptors. And now This is the afferent arteriole, and these are the granular cells. Granular cells, it means that they must have some granules in them, and they contain granules which contain renin. And we know renin is very important in blood pressure management. And these cells, they act as the mechanoreceptors. When the blood pressure is when there is increased or decreased blood pressure, they can detect that. So let me label them. These are mechanoreceptors. These cells, which are the granular cells, they are mechanoreceptors. They can detect the blood pressure. And then in between these two cells, you can see a third type of cells which are known as mesangial cells. So these are mechanoreceptors. Alright, then these are the three types of cells, which are the mesangial cells. And these mesangial cells, they have plenty of gap junctions in between them, so substances can pass between these cells. So this apparatus is known as juxtaglomerular complex of the nephron. And I'm sorry about this, I didn't write it very neatly. This is all about the anatomy of the nephron, and you can go to this link and you can watch our video to have a clear picture of everything.